Okay, thank you, and good evening, everybody. First of all, let me thank the organizers for giving me this opportunity. So, I would like to talk about the possibility to use a sort of multimodal camera. Suppose I have to make a photo here, of all of you, and I want to catch all of you in the same moment. I want to have the possibility to zoom in and to see every single cell of your body. What I want to demonstrate to you today is that I have to use a multimodal camera, which is a sort of, let's say, correlative microscopy approach. Now, the old features that you experience in the brain, like the movement coordination, the organization of sensory data, the memory, or the creativity of the abstract reasonings, relies on a local behavior at the cellular level, but also on the organization of those local behavior between themselves on a long-range projection. And the hard task is that in the brain there are 10 to the 11 neurons and 10 to the 15 synapses, and even if you go to mouse brain, you have an enormous amount of neurons and synapses. Now, the anatomical structure of the brain spans from a few hundred of nanometers from synapses, and you have to measure and visualize them, but then you need to measure and visualize the dendritic arbors, and then you have to measure and realize the connections between all neurons in the whole brain. And so, structure, functionality, and plasticity are three important key features in the brain that must be studied and connected between the morphology and the functionality all over the brain. And one point, for example, and the one key aspects you can study in the cerebellum, for example, is how Purkinje cells are ruling the deep cerebral nuclei, because they are inhibiting the deep cerebral nuclei and they are controlling the motion activity. And they are connected to the inferior olivary by climbing fibers and to other connection by parallel fibers. And they are driving exactly the behavior of the Purkinje cells. Now, the plasticity of those climbing fibers and parallel fibers are very important for the behavior and the motion. And what's the way, in that case, to study the plasticity? And okay, the optical manipulation now it's expanding on two big fields. One is optogenetics. The other one is the possibility of using nonlinear interaction of the laser light to perturb the systems. In this case, is talking about laser axotomy. And here, in vivo, by using a cranial window systems, then you can dissect in a very precise way some climbing fibers. In this case, we dissected 25 climbing fibers, okay? And then we studied, after a few days, the formations of new branches. And here you can see here a new sprouted branch which was formed. Now, the problem is the following. Is this branches forming a new synaptic contact? So, if I'm using two-photon microscopy, as I'm doing here in vivo, I'm not able to establish the contact because the resolution is not enough to do that. So, what I have to do is, you have to pass to another techniques, which is, at that point, electron microscopy. So with focus ion beam, I can, at that point, I see in vivo the brain, excise the brain, fix the brain, slice the brain, but I have to find the same region. And to find the same region is just like to find a needle in all these big buildings. So the way to visualize exactly the same region is using laser writing. In that case, we make a mark all around the sprouted branch, okay, and that mark was used to be found again with electron microscopy. And then we found a fantastic thing, is that the sprouted branch in green was adapting completely to the shape of the dendritic branch. And then all this adapting at that point the question was, is it going to establish a new synapse formation? And then if you zoom in, with electron microscopy, you see that all the machinery to create a synapse is present. So combining a two-photon and correlative and electron microscopy, it is possible to zoom in and to see if the perturbed systems and the reactive plasticity is forming new branches, is forming new synapses. Now, the problem is that the local plasticity is connected to long-range connections. And two-photon is not available to go down up to the 5th, 6th layer in vivo. And electron microscopy is too much consuming to visualize connection all over the brain. So the answer to that is to correlate those two kinds of microscopy to a third kind of microscopy, which is the light sheet microscopy. Light sheet microscopy is always on ex vivo sample, but illuminate with a plane of light the brain, which is visualized at 90 degrees, then the light is moved all over the brain, and the 3D tomography can be obtained. Of course, the brain has to be cleared, and by removing the water with the fluid index matching, you can make the brain completely transparent. There are different kinds of approaches, and recently Clarity seems to be interesting because it allows also the post-mortem labeling of the brain. In any case, we developed a new method which combines the light sheet microscopy with confocal detection, which allows to remove the scattering which still remains into the brain and visualize single processes well within the brain without slicing the brain. And doing that, we reconstructed the whole brain, which was possible to be zoomed in different regions and tracing single processes, and develop a new software, Google Brain, which was inspired to Google Maps and to Street View. In that case, it is possible to zoom into the brain, to navigate, and to change all the different direction and perspectives. But also, we developed in this terabyte-size data a new software to count cell by cells. This is, for example, the counting of all the cell, and for each cells we know the X, Y, and Z position. Of course, by using some animal model, it is also possible to visualize the activated regions after a task. And the interesting thing is that we can visualize activated regions cell by cell with a resolution which is about 1,000 times higher than the nuclear magnetic resonance. Of course, all this data must be connected with the vasculature connections and morphology, as was shown in the previous talk. Now, let me say at that point that we have to correlate the two-photon with the light sheet microscopy. So first we make the two-photon, then we excite the brain, and then we make the light sheet. And in order to correlate that, first of all, we correlated with the two-photon with the light microscopy. We illuminate with green light in order to have a very high contrast on the blood vasculature. The blood vasculature is a fingerprint, so we could correlate two-photon with light microscopy. And then when we make a light sheet microscopy, also the perfusions which is used is also fluorescence, and also create a blood vasculature pattern so we can correlate the two. At that point, we can visualize the volume where the two-photon dendrite was visualized within the whole volume of the brain, and we can correlate the plasticity at the dendrite level with the long-term projections. This is very interesting because it allows us to exploring the brain on complementary spatiotemporal scales. Two-photon is limited in space, but not in time. So confocal light sheet is limited in time, but not in space. Enable us to correlate single neuron functionality, plasticity with its morphology and connections, and tracking, migrating, and differentiating cells in correlation with the overall 3D cell distribution. Studying plasticity brain rewiring after brain injury or sensory deprivation, and discriminating large volume, the sprouting dynamics dependence from neural type. Let me say that this work was done in the frame of the Human Brain Project, which is the European flagships the twin project with respect to the brain, American one. In this case, it's a 3 years project, and in the frame of this project, we are coordinating our institute, the optical imaging part. Fiber optics is transferring the data to high-performing computer centers, and all scientists are working in remote on this data by plugging instruments and software to measure, to visualize, and to navigate into the brain. Let me thank the people, first of all, the young collaborators of my group, but then the people in the other institute of Europe. Let me say that strong are also the connection with the United States, with the Allen Brain Institute, Medical Harvard School, and the University of Connecticut. And let me also thank you for your attention for this journey into the brain.
Thank you very much. This concludes our hot topic session. I would really like to thank all of the speakers.